Hi, hello everybody. My name is Gustavo Adolfo Cardoso. Today is June 23rd, 2021, and this is my Unit 4 case connection video. So for this assignment, I chose case number 1 and number 2. So starting with case 1, it talks about a newborn girl who appeared normal at birth, but within 24 hours later, she developed lethargy, hypothermia, and apnea. The attending physician suspects metabolic disorder and orders a panel of labs. Blah, blah, blah. So the initial blood ammonia level was elevated at 314 micrograms per deciliter, and the normal range is 17 to 80 micrograms per deciliter. And it also says that it rapidly rose to 600 micrograms per deciliter. It's asking to discuss the mechanisms of ammonia transfer in the body, and be sure to include glutamate dehydrogenase, glutaminase, and glutamine synthetase. It is also asking to discuss how nitrogen transport is connected to gluconeogenesis in the fast state, and it provides It also asks to discuss how nitrogen transport is connected to gluconeogenesis in the fasted state. And then there was a further analysis showing that uric acid was elevated at 473, and the plasma citrulline level was normal at 10. Normal levels are 12 to 55, so it's a little bit under. Okay, so protein and amino acid metabolism are a key component to the formation of ammonia. Besides being important building blocks to supply nutrients for the maintenance and growth of cells and tissues, protein and the resultant amino acids from its breakdown play an important role in providing substrates for nitrogen and carbon-containing compounds that assist with neurotransmitters and nucleotides, as well as in fatty acid synthesis and gluconeogenesis, respectively. However, excess nitrogen must be disposed through a series of transamination reactions and oxidative deaminations in the liver via the urea cycle. Any dysfunction of the cycle disrupts nitrogen homeostasis and may result in the accumulation of ammonium ion, NH4+, which is highly toxic in the body. Urea biosynthesis occurs through a series of 4 reactions: transamination, oxidative transamination of glutamate, ammonia transport, and reactions of the urea cycle. So there are 2 primary nitrogen carriers in the body: alanine and glutamine. Alanine is transaminated to alpha-ketoglutarate by alanine aminotransferase, which produces glutamate. Glutamate is fixated with ammonium by glutaminase synthetase, producing glutamine. This occurs in most tissues of the body. Glutamine is then mobilized to the liver, where it's deaminated by glutamase, freeing NH4 or ammonium, and regenerating glutamate. This free ammonium enters the urea cycle, while the newly formed glutamate compound undergoes a reversible reaction with glutamate dehydrogenase, which, along with cofactors NADPH and NADH assistance, it produces alpha-ketoglutamate and releases a final ammonium ion that enters the urea cycle. So the nitrogen transport is connected to the gluconeogenic pathway through the alpha-ketoglutarate generated in the process, because it serves as a substrate for gluconeogenesis and fatty acid synthesis that enter the TCA cycle and can be converted to oxaloacetate, which enters the cycle. So nitrogen transport is connected to the gluconeogenic pathway through the alpha-ketoglutarate generated in this process, because it serves as a substrate, as I already mentioned, for gluconeogenesis as well as fatty acid synthesis that enters the cycle and can be converted to oxaloacetate, which also enters the cycle, preceded by the enzyme alpha-ketoglutarate dehydrogenase complex, similar to pyruvate dehydrogenase complex. So alpha-ketoglutarate dehydrogenase complex is one of the 2 regulatory enzymes of the cycle. It is also connected as ammonia accumulation leads to a depletion of the oxidative decarboxylation alpha ketoglutarate, which impairs the function of the TCA cycle. So urine orotate or uric acid comes from the alternative pathway that accepts carbamoyl phosphate, CAP, which is also synthesized in the cytosol by CPS2 in pyrimidine synthesis to produce orotate. So based on this, it is understood that excess accumulation of orotate results from the urea cycle deficiencies, particularly of ornithine carbamoyl transferase, or OTC, as this enzyme catalyzes the reaction between CAP and ornithine to produce citrulline in the mitochondria. So this deficiency promotes CAP to be subsequently used in pyrimidine synthesis, yielding one of its intermediates, which is orotate or uric acid, in overabundance. So this explains why there is a reduced level of citrulline observed in further analysis. Again, 10 versus 12 to 55, which is the normal, a bit reduced. So considering that there is an OTC deficiency which leads to the underproduction of citrulline, it means that it would lead to a deficiency of both the urea cycle and the production of amino acid arginine. This is because cleavage of arginine releases urea, or NH2, and reforms ornithine by enhancing N-acetylglutamate required for CAP generation through CPS1. Okay, so moving on to case number 2, it provides 3 different scenarios, and it's asking to discuss 3 things. Number 1, the most likely enzyme deficiency in which amino acid or amino acids are no longer metabolized. Number 2, if any amino acid becomes essential as a result of a deficiency, discuss it. And number 3, discuss potential vitamin supplements that might be useful for the condition, and what steps in the metabolism they could potentially help with. All right, so scenario number 1 is talking about a full-term girl was born by normal vaginal delivery and was released from the hospital at 3 days of age. At 9 days of age, a screening test for branched amino acids came back positive, and the child was readmitted to the hospital for further evaluation. There was no history of genetic disease in either family, and both older siblings are well. Branched chain amino acids, or BCAA metabolism, begins with its transamination, just like with any other amino acid, to a keto acid that allows the removal of the nitrogen group. So the newly formed keto acid undergoes an oxidative carboxylation by branched chain alpha keto acid dehydrogenase, or KDCA, which is an enzyme similar to pyruvate dehydrogenase, or PDC. The remaining carbon skeletons are rearranged and enter the TCA cycle. I believe that there is most likely a deficiency in the amino acid thiamine, as it is required for the proper functioning of KDCA, just like in PDC. So KDCA deficiency is also quite possible, as there is a downregulation of BCAA metabolism, and action of the enzyme is favored by thiamine. For this reason, thiamine diphosphate in its active form, B1, vitamin B1, should be administered to the newborn patient. Okay, scenario number 2, it talks about a 69-year-old woman who's got a 30-year history of low back pain and large joint pain of the hips and shoulders, and on examination, they noticed a blue-gray pigmented macules that were present over the cartilaginous portions of the ears and on the sclera. Past medical history includes aortic stenosis. Okay, so this patient most likely has a deficiency in tyrosinase enzyme, which is the rate-limiting step involving tyrosine metabolism. So tyrosinase converts tyrosine into dopaquinone, which is then used to generate melanin. So the inability to generate melanin as a result of this deficiency in tyrosinase results in hyperpigmentation, like the one observed in the patient. Tyrosinase can undergo a separate pathway in which its final products are fumarate and acetoacetate, both of which may enter the TCA cycle. Acetoacetate is particularly important here as a deficiency in its metabolism, especially at homogeneous oxidase, the rate-limiting enzyme responsible for catabolizing homogeneous so that it can be further modified into acetoacetate and fumarate, promotes elevated levels of this metabolite, homogeneous. Elevated levels of this metabolite are spontaneously converted to alkapton, leading to a condition referred to as alkaptonuria. Alkaptonuria is a condition marked at first by the darkening of urine when exposed to air. Eventually, the condition evolves to arthritis and connective tissue pigmentation or ochronosis, in particular at the nose, cheeks, forehead, hands, eyes, neck, most of which kind of like coincides with those observed in the patient. Treatment for this condition is limited. However, homogeneous acid excreted can be partially reduced with dietary restriction of tyrosine and phenylalanine. Treatment for this condition is limited. However, homogeneous acid excretion can be partially reduced with dietary restriction of tyrosine and phenylalanine. And this is because hydroxylation of phenylalanine by the enzyme phenylalanine hydroxylase results in the reduction of tyrosine. Okay, so moving on to the third and last scenario. This is about a 4-day-old infant who was born through vaginal delivery, had a normal pregnancy, and her birth weight was 5.9 pounds. They did some screening and demonstrated that phenylalanine levels were 6.6 milligrams per deciliter, when the normal range is 1 to 2 milligrams per deciliter. So it's very elevated. Okay, so phenylalanine is an essential amino acid that participates in a series of reactions that are necessary to the overall metabolism. Its metabolism is responsible particularly for the generation of tyrosine, a non-essential amino acid that provides substrates for the synthesis of thyroid hormones, T3 and T4, as well as the generation of melanin, as we discussed just previously. So elevated levels of phenylalanine are caused by a deficiency in the rate-limiting enzyme, phenylalanine hydroxylase, or PAH, or in the cofactor tetrahydrobiopterin, or BH4, which is required for proper functioning of the enzyme. Deficiencies in either PAH or BH4 result in phenylketonuria, or PKU. Impaired PAH functioning makes tyrosine become a conditionally essential amino acid. So PKU is a hereditary autosomal recessive disorder characterized by a deficiency of PAH or its cofactor BH4, like we just discussed. So this deficiency causes the accumulation of phenylalanine in body fluids and in the central nervous system, which can be bad. Phenylalanine is metabolized to phenylketones and is excreted in urine. So given that tyrosine becomes conditionally essential in the patient with PKU, and with the understanding that is a newborn patient, supplementation via formulas rich in tyrosine may assist with the metabolism of this important amino acid. That is it. That's the end of the semester.